Mic check.
Mic check.
Hello, everyone. Welcome to Antibuddies. This is our 31st Journal Club, and we have a special guest today with us, Dr. Jan Rehwinkel. He is a professor at the University of Oxford, UK, and I will let Dara tell us more about our guest today.
Thanks, Jatin. So let me first introduce our today's guest, Dr. Jan Rehwinkel from the University of Oxford. So his lab investigates the molecular biology of nucleic acid sensing and its relevance to human disease. So nucleic acid sensing is basically a fundamental process in the immune system. Mammalian cells, including immune cells and non-immune cells alike, are equipped with sensors for these unusual DNA or RNA. And in today's journal club, he's going to be giving us an example of the double-stranded RNA adopting the so-called Z conformation and its role in innate immunity. Outside the lab, Jan enjoys spending time with his family and his 2 children, as well as long-distance running. Thank you once again for joining us today, Jan.
Great. Thank you very much, Jatin and Dara, for the kind introduction and for the invitation to present today. And thanks to all of you for coming out on a Saturday, I guess morning in the US and afternoon in Europe. So I appreciate that. So what I'd like to introduce to you today is an unusual form of nucleic acid called Z. And I should say up front that I'm not a native speaker, but I've lived in the UK for almost 15 years now. So I will use Z and not Z. So I apologize for that, but I've got very used to the British pronunciation. So I'll tell you about these unusual Z nucleic acids. But before getting into that, I wanted to start with a bit of a broader overview of my wider field of research, and that is nucleic acid sensing by innate immune receptors, as Dara has said. And really the key observation in this field is that nucleic acids, both DNA and RNA, are very potent triggers for innate immune responses. And that can happen in many different disease contexts. That, for example, it includes infections, in particular viral infections, but also many different settings of sterile inflammation, including some autoinflammatory diseases such as lupus, but also in some instances malignant diseases. And what's happening in all of these different settings is that unusual nucleic acids accumulate, and we call these immunostimulatory DNA or RNA. And in the context of a virus infection, it's often quite simple in that viruses have either a DNA or an RNA genome in their particle, and they deliver that genome into the cell that they infect. And then that's recognized by the innate immune system as an immunostimulatory nucleic acid. In settings such as cancer or autoinflammation, it's often more complicated. For example, in cancer, this sometimes goes along with genome instability, and that generates DNA fragments that then can also become immunostimulatory and are recognized by the innate immune system. And that event of recognition is mediated by a very large family of germline-encoded receptors or sensors that we collectively call nucleic acid sensors. And here just a few examples that are very widely studied, such as the toll-like receptors, the DNA sensor CGAS, or the Rehwinkel receptors, which are receptors for RNA. But it's important to point out that these are just examples, and there are many more of these types of nucleic acid sensors. And there's also a large a variety of outcomes, signaling outcomes that these sensors trigger. And amongst those different cellular responses are the production of cytokines, in particular type I interferons, which are cytokines that are really important for antiviral defense. Another important signaling outcome once these nucleic acid sensors are activated is the induction of programmed cell death, whereby the cell in which these unusual nucleic acids accumulate effectively commits suicide and undergoes apoptosis or other forms of programmed cell death, and that, for example, can stop a virus replicating in the cell that it's infected. So this is sort of a high-level overview of the process of nucleic acid sensing. And if you think about this from a very sort of fundamental point of view, then there is a problem. And the problem is that any normal cell that's not infected, that's completely healthy, already contains a lot of nucleic acid. There's DNA in the cell nucleus and a bit of DNA in mitochondria, and there's plenty of RNA in most compartments in the cell. So that raises an interesting question, and that is how can these innate sensors distinguish between the normal nucleic acid content in a healthy cell and unusual or pathogenic nucleic acids that might accumulate in a cancer cell or in a virus-affected cell or in a cell that initiates an autoimmune response. And it's that fundamental molecular question that my lab is interested in answering. And as it turns out, there are a number of different molecular and cellular solutions that have evolved and that have been described in our field. And the first of these solutions is the recognition of nucleic acids in unusual places. And one example for this is this protein called CGAS, which is a DNA sensor. And now CGAS is active in the cytoplasm of cells. Now, the cytoplasm of cells usually does not contain DNA. DNA is contained in the nucleus, and a bit is also contained in mitochondria, but the cytoplasm itself is usually in a normal cell free of DNA. But if there's a problem, such as a DNA virus infecting the cell and delivering a DNA genome into that infected cell, then CGAS is recognizing the DNA in the cytoplasm and activates an interferon response and other innate immune responses. So one sort of cellular solution is detecting nucleic acids if they appear in the wrong place in the cell. Another molecular solution that has evolved is the recognition of the presence or absence of chemical modifications that might, for example, be found or not found on viral nucleic acids. And a good example for that is the recognition of some viruses, such as influenza virus, by a protein called Rehwinkel, which is an RNA sensor that also induces an interferon response. And what happens here is that the flu virus introduces its genome into the cell, which is an RNA genome, and these flu RNAs are not very much processed. And in particular, they're not kept at the 5' end. And that is a little molecular difference that allows Rehwinkel to detect these flu RNAs, because most of the cellular RNAs, for example, messenger RNAs, are kept. And that's not happening with these flu RNAs. And therefore, that allows this particular innate immune receptor to recognize this difference in chemical modification on the RNA from the virus in this case. Yet another solution for this problem of distinguishing normal and unusual nucleic acids is the recognition of specific nucleotide sequences. And there are examples for that in the field of toll-like receptors. And then finally, there's yet another solution, and that is about secondary structures that nucleic acids adopt. And I've highlighted this in blue here because that's what I'd like to cover in the journal club today. And so let me move on then to introducing to you Z nucleic acids. Now, if we think about the secondary structure of nucleic acids, then this is the image that comes to mind, the double helix of the DNA that everyone learns about at university and perhaps even at high school. And so the important thing to note, though, is that this is not the only conformation that a double-stranded nucleic acid can adopt. There are other types of conformations, and here is one of them. which is called Z. As you can see, it looks very different. And some of the fundamental differences are that the bDNA, the conventional double-stranded DNA, is a right-handed double helix, whereas zDNA is a left-handed double helix. In addition, the structure of the phosphodiester backbone, which is this black line in these cartoon here, is quite different. In bDNA, you've got this sort of smooth backbone that runs around the helix, but in zDNA, this has a zigzag shape, and that's actually why this conformation is called zDNA, because of the zigzag phosphodiester backbone. There are some other differences, such as the absence of major groups in zDNA, as well as the orientation of the bases, where some bases are actually not facing inwards, but are pointing outwards. And this unusual structure of DNA was discovered in the late 1970s by Alexander Rich, shown here through structural analysis. And shortly after, others showed in the 1980s that double-stranded RNA can also adopt the unusual double-stranded left-handed conformation. And so there's both zDNA and zRNA. So what do we know about these unusual nucleic acids? Well, the first thing to note is that they're not very stable conformations. And if you look at the thermodynamics, then in the normal physiological buffer, really the B conformation of DNA, or it's called A conformation RNA, these classical conformations are really preferred. And the Z conformation is not preferred. However, certain repetitive sequences such as DG, DC, repeating dinucleotides, can spontaneously switch into this Z conformation in very high salt buffers, such as 4 molar sodium chloride. So under non-physiological conditions, very high salt buffers can induce this conformation. In addition, there are other chemical tricks that allow the stabilization of this form of nucleic acid. And that in turn, again, a long time ago, allowed people to raise antibodies against both zDNA and zRNA. And these antibodies were quite informative in that using them for immunofluorescence suggested that cells contain nucleic acids in this conformation. So these anti-zDNA antibodies mostly stay in the cell nucleus, and the zRNA antibodies stay in basically all of the cells. So potentially these nucleic acids exist, or at least there's some nucleic acid that can be stabilized in this conformation through binding to these antibodies. I see there's a question here, so I'll briefly pause and take a question.
Yeah, sorry. I have a question because I was wondering, is it like the whole DNA or the whole RNA has this zDNA structure at the time, or is it like it can only be like fragments, for example, like, I don't know, 50 base pairs or something?
Yeah, that's a very good question. And those experiments that were done in the early days in the 1970s and 80s, they usually involved only short oligonucleotides, where the idea is that the whole oligonucleotide of a very short length would switch into Z conformation. However, in longer stretches of DNA, there is something that's called the B to Z junction, where basically one conformation flips into another. And so the idea is that in longer nucleic acids, there might be stretches that adopt the Z conformation. And that's actually quite a good point, because it leads me on to the next thing that I was going to tell you about, which is biological functions of these. And that's really still an area of active investigation. And for zDNA, the idea is that this conformation relieves torsional strain induced by the movement of polymerases. So if you imagine a double helix of the DNA, and then you imagine a DNA polymerase or an RNA polymerase replicating or transcribing a stretch of this DNA, then it has to unwind the two strands of the double helix in order to copy one of the strands. And so if you imagine you've got sort of two bits of rope coiled around each other in a double helix, and then you pull those two ropes apart in order to get at the basis of one of them, then you induce some kind of supercoiling ahead of that point where you pull them apart. And the idea is that this torsional strain is released by flipping into the Z conformation before and behind transcription bubbles or replication bubbles, and that releases that torsional strain that's introduced by polymerase basically pulling the two strands apart to transcribe or replicate one of the strands. And so in that sense, the idea is that, let's say, in an intact chromosome, if we speak about Z DNA, that there are stretches where B is flipping into Z to relieve torsional strain. Jatana, you've got a question?
Yes, just a very basic question. The last time I learned about ABC conformation was in my undergrad, and I never thought I would ever use it until today. So, are there specific polymerases that act on Z DNA, or are they even transcribed?
I don't know the answer to that. To the best of my knowledge, there's no specific DNA or RNA polymerase that would transcribe or replicate Z DNA. I think generally this conformation is probably something that happens quite transiently and is only in a very small part of the overall genome.
Okay.
So I'm not aware of a polymerase that would specifically bind to Z. Right. So as for zRNA, but when we started this work a few years back, biological functions were entirely unknown. However, there is an important clue, and this clue is that some proteins have a domain called Z alpha, and you see down here the structure of this domain. This is from an old paper, again from Alexander Rich's lab from the late 1990s, where they've solved the structure of one of those Z alpha domains, shown here in contact with zDNA. And this domain is really short. It's only about 80 amino acids long. And it very specifically binds to both zDNA and zRNA and doesn't bind to aRNA or bDNA. And what us interested is that there's only a very small number of proteins, in fact 4, that have these Z alpha domains. And interestingly, all of these proteins are involved in innate antiviral immunity. And so the first of these proteins is a protein coded in mammalian genomes called ZBP1 that I'll briefly describe on the next slide. But I just wanted to go through the list of those proteins here. The second one is ADAR1, and I'm going to spend most of my time talking about it. Again, it's encoded in mammalian genomes. And then we've got these two proteins here. The first one is encoded by vaccinia virus, which is a pox virus. And this protein is called E3L, and it's got one of these domains. And this E3L protein is an antagonist of innate immunity and allows pox viruses to evade the innate immune system. And then finally, there is a protein from zebrafish called PKZ that has two Z alpha domains and a kinase domain. And this protein is very similar to a mammalian protein called PKR, which is involved in shutting down translation during virus infection. So it seemed that all of these proteins that have the Z alpha domain were in one way or another involved in innate immunity. And so therefore we thought that the function of zRNA would probably be somewhere in the field of innate antiviral immunity and nucleic acid sensing. And so therefore we started to investigate this. And I just wanted to very briefly highlight some work that we've published on this protein called ZBP1, which came out about 5 years ago. And because it's a bit old, I'm not going to show the data, but I just wanted to highlight what we and others had found at the time. And this was that during infection with viruses, we used a mouse cytomegalovirus, a herpes virus, zRNA is introduced into the infected cells. And this is then recognized by this protein ZBP1, which has 2 of those Z alpha domains in its N-terminal portion. And this binding to zRNA then activates ZBP1, which will then activate various downstream signaling pathways. And one of these is shown here, where a kinase called RIPK3 gets activated. That phosphorylates a protein called MLKL. And this MLKL protein is a suicide protein that upon phosphorylation inserts itself in the plasma membrane and forms a pore. And then cytoplasmic content is leaking out of the cell, and the cell is swelling due to the influx of water. And then basically cell explodes and dies. And this form of programmed cell death is called necroptosis. And the model is that this is beneficial to host defense and innate immunity, and that the cell recognizes it's been infected and then it sacrifices itself and dies by programmed cell death, and thereby prevents replication of the virus in that cell. And as a note on the side, this MCMV encodes a protein called M45, which is a viral antagonist of this process. And this M45 protein blocks the interaction of ZBP1 and RIPK3. So you can make the virus a lot more susceptible to this by mutating or deleting the M45 protein. Now, others who worked on this as well, in particular Sid Balachandran's lab, found that the same is happening during a flu infection, where flu also introduces Z nucleic acids, and the same, as shown by Mokarski's lab, happens during poxvirus infection. And in addition to those viral settings, there are a number of settings where self-RNA in the Z conformation builds up during autoimmunity and autoinflammation, and also triggers a ZBP1 dependent cell death process. So these data established zRNA as a molecular signature that's read by the innate immune system, in this case by ZBP1, to induce programmed cell death and other signaling outcomes. Now, because this work has been published quite a long time ago, and because you so specifically invited me to speak about our work on ADAR1, I'll move on to that swiftly and tell you about this protein. So ADAR1 is an abbreviation for adenosine deaminase acting on RNA. And this protein comes in two isoforms. There is a longer isoform called P150, which has a Z-alpha domain near the N-terminus. And then there's a shorter form called P110 that's lacking this N-terminal portion, but is identical in the C-terminal portion. And what these two isoforms have in common is at the C-terminus, this deaminase domain shown here in orange. And this domain has a catalytic function, and this activity is to deaminate adenosine to inosine in RNA. And that's chemically shown here, where this adenosine is changed here to an inosine in this deamination reaction catalyzed by the deaminase domain in ADAR1. And this process is called RNA editing, and that A's are converted to I's. There are thousands, if not millions, of sites that are edited in the human transcriptome. And most of these sites that are edited are in non-coding RNAs and in a repetitive RNA. And this is important because A2I editing destabilizes double-stranded RNA structures. And that is because adenosine pairs with uracil in a double-stranded RNA, as shown on the left here. However, if this adenosine is edited to an inosine, then the base pairing properties change, and the inosine will pair with cytosine, as shown here. So if you imagine a double-stranded RNA with multiple A's that pair with opposing U's, and then these A's get edited to I's, then the stability of this double strand will be disrupted because the I doesn't have the appropriate base to pair on the opposite strand. Now that in turn is important in human disease, and it's now quite well established both from human genetics, particularly in the context of a disease called Aicardi-Goutieres syndrome, as well as from mouse models, that ADAR1 deficiency results in interferon-driven inflammatory disease. And so in the human setting, this rare disease, Aicardi-Goutieres syndrome, has some overlap with lupus. It's a very, very severe disease that's driven by spontaneous production of interferon in the absence of virus infection. And it can be caused, amongst other mutations, by mutations in the gene encoding ADAR1. And that can be modeled in mice. If you knock out the entire ADAR1 gene in mice, then these mice are embryonically lethal. They will develop a little bit, but then in utero, the embryos are resorbed. And before that happens, those embryos have a very, very strong type 1 interferon signature. Now, what mouse genetics by a number of different labs, and the citations are down here, has shown is that an RNA sensing pathway drives this interferon induction. And this RNA sensing pathway involves MDA5 as the RNA sensor and MAVS as the adapter protein. And this drives interferon inductions in the absence of MAVS. If you knock out either MDA5 or MAVS on top of ADAR1, then a lot of the problems that these animals have are rescued, and showing that this is what initiates some of the downstream disease. And so this leads to this model shown here on the right, where some form of endogenous RNA needs to be edited by ADAR1. And if that doesn't happen, then this RNA activates MDA5, which then it triggers MAFs, and that activates a downstream signaling pathway involving kinases such as TBK1 and transcription factors such as IRF3. And then you get expression of interferons and antiviral genes. So this was all quite well established, but the field had paid much less attention to how the RNAs are selected by ADAR1 for editing. And the protein has multiple domains that recognize nucleic acids. It's got these double-stranded RNA binding domains here, but the long isoform of ADAR1 also has the Z-alpha domain that I've introduced a few minutes ago. And because we were quite interested in those Z nucleic acids, we decided to investigate the role of the Z-alpha domain in ADAR1 in spontaneous interferon production and autoimmunity. And so what's shown here, again from this old paper of the structure of the ADAR1 Z-alpha domain, is a blow up where you, again in blue you see the Z-alpha domain, and then in the red colors a bit of Z DNA. And I wanted to start by highlighting this particular amino acid, the proline, at position 193. And this is an interesting position because some patients that have Aicardi-Gouttière syndrome, this interferon-driven disease, have a proline to alanine mutation found at this position. So that indicates that this domain is probably quite important to the disease. Now, to study its role, we wanted to take a mutagenesis approach, but we were quite interested in understanding also the importance of binding to zRNA. And what we decided to do, therefore, was to mutate to different amino acids. This is paragene at position 173 and this tyrosine at 177. And the reason that we chose those was that previous biochemical and structural studies by others had really shown that these two are essential for binding to Z nucleic acids. And so what we did was to mutate those two into alanines in a mouse background. So these are knock-in mice that instead of the normal ADAR1 gene have an ADAR1 gene where these two amino acids are mutated to alanine. And this here is a sequencing trace to validate that we've made these mice correctly. So on the top here, you've got the sequencing trace of wild-type mice. For example, look at this position, we've got AAT, two green and a red peak encoding asparagine. And if you look down here, we've got a black, a blue, and a red peak. So that's GCT, and that's alanine. So that validates that we've introduced these mutations into the germline of these mice as we had predicted. And the first observation that we then made when we looked at these animals was that these ADAR1 mutant mice, and I'm going to call them ADAR1 MZ alpha for short in the rest of the talk, were born at predicted Mendelian ratios, developed normally, and were fertile. And so that's quite different to the full body knockout of ADAR1, which is embryonically lethal, suggesting that really the Z alpha domain is not absolutely essential to the function of ADAR1, but it was sort of a good result for us at the time because it allowed us to study these mice and to breed them and to look what's going on. And so the first observation that we then made looking at these animals was that if we collect RNA from different organs, in this case, lung, liver, and spleen, that there's actually evidence for the production of interferons in these different organs. So what we've done here was to extract RNA and then do an RT-qPCR for the transcript encoding interferon beta, one of the type I interferons, or for the transcripts encoding IFN1 and IFI44, which are two interferon-stimulated genes that play roles in antiviral defense. And the color coding is such that blue is wild type and red is mutant mice. And you can see that in the organs that we've analyzed, interferon-stimulated genes were upregulated in our mutant mice compared to wild-type mice. And this induction of an interferon response was specific in the sense that other markers of inflammation, such as type 2 interferon gamma, TNF-alpha, or IL-1 beta, these different transcripts were all more or less unchanged in the mutant mice compared to our wild-type mice. So this suggested that these animals show spontaneous production of type I interferons, and we've validated this in multiple ways. One is shown here, where we've done an RNA-seq on lung RNA, and then looked at the transcripts that were upregulated in the lungs of the mutant animals. And if we do gene ontology analysis, then the upregulated genes are involved in things such as defense response to virus, interferon beta, etc., negative regulation of viral genome replication. And indeed, if we look at the set of upregulated genes, then many of these are classical interferon stimulated genes. This is probably too small to read, but there's things such as MX1, ISG15, IFIDs, etc., that are classical interferon stimulated genes. So this validated that these animals, despite being overall normal, produce more type I interferons than a wild type mice would do. So that it suggested that the Z-alpha domain plays an important role in recognizing RNAs that need to be edited to prevent interferon responses. And what we were also interested is whether these mice might be protected against virus infection. And as I've mentioned, interferons are really potent cytokines against viruses. So if there is a bit of an interferon response going on in these animals, and if that's functional, then the prediction is that they might be protected against virus infection. And so to do that, we used influenza virus as a model, and we've administered the flu virus intranasally at a sublethal dose, where wild-type mice, again shown here in blue, would start lose some weight around day 3, and then they would continue to lose about some weight up to 10% after about a week after infection, and then these mice after that would recover, clear the virus, and gain weight back to normal. Now, if we compare this weight curve, which is a good readout for what's going on in these animals in terms of pathology and virus replication, if we compare that to our mutant mice, shown in red, then it was interesting that around day 3 and day 4, when the wild-type mice already started to lose weight, these mutant mice were not losing any weight, and the weight loss only started later, and then they would also lose around 10% and recover. So that suggested that at this early stage, these animals might be doing better and might be protected by that interferon that's going on at baseline. And indeed, when we looked at the lung index on day 3, so at this time point here, with the lung index being a measure of sort of pathology in the lung, then this was higher in our wild-type mice compared to our mutant mice, suggesting that the mutation protects. And if we look again at this early time point of day 3, at viral RNA as a readout for viral replication, then we found that pRNA and mRNA, which are two of the viral genomes, genome segments of flu, again, there was more viral RNA in the wild-type mice in blue compared to the mutant mice in red. So that suggested that this interferon response that's going on in these ADAR mutant animals was not only detectable by qPCR, and other means like sequencing, but was also in fact functional and partially protected against flu at an early stage of the infection. So then next, what we wanted to do was to understand which signaling pathways might be driving this interferon response when the Z-alpha domain of ADAR1 is mutated. And given the previous work that the RNA sensor MDA5 and the adapter protein of it called MAVs are involved in interferon induction in the full ADAR1 knockout, we hypothesized that the same might be true in the ADAR mutant mice that we generated. And to test this, we crossed our mice to MAVs knockout mice, so taking out this adapter protein here. And it's the same color coding that you've seen before, but now we've got green, which is MAVs knockouts, and then we've got purple, which is the double transgenic mice, mutant for ADAR1 and knockout for MAVs. And here is RNA extracted from brain and RT-qPCR data, and we've got the very similar data from other organs. And again, what we find is that when we compare wild type to mutant, that there's an increase in interferon beta and different interferon stimulated genes. So that's red compared against blue. However, if we look at the mutation in a MAVs deficient background, so now comparing purple to green, there's absolutely no increase happening, showing that this interferon response that we observe is dependent on MAVs, and we believe very likely on MDA5 as well. And then finally, the last sort of set of experimental data that I want to share is about RNA editing in these animals. And we had sort of two different ideas here. The first was that maybe the mutation of the Z alpha domain has a global impact on editing and somewhat reduces editing at all sites where RNA editing occurs, maybe by 10 or 20%, and that results in this sort of moderate interferon response that we observe. And the other model was that perhaps there's a subset of editing sites where editing is really severely affected, whilst at many other sites, editing is completely normal in these mice. So that there might be a smaller number of sites where the Z alpha domain is really important for editing. And luckily, we were able to analyze RNA editing in our RNA-seq data. And wanted to take a minute to briefly explain how that works. So if you've got an RNA that you extract from cells or from tissue, and then you do an RNA-seq, one of the things that first happens is that the RNA is reverse transcribed into DNA, and then it's actually the DNA that's sequenced. And so during reverse transcription, a reverse transcriptase will copy the RNA into DNA. And I've mentioned earlier that A pairs with U, but I pairs with C. So if you imagine the reverse transcriptase, if it encounters an A, so non-edited, then it will put a T in this position. However, if reverse transcriptase comes to an I, so to an edited position, then it will put a C because I pairs with C. If you then have that cDNA and that then gets sequenced, then the T that's put here by the reverse transcriptase will be read out as A as expected, but the C here will be read out as G by the sequencer. And so if you look at RNA-seq data and you look at deviations from the genome annotation at mutations, then A to G transitions really happen quite frequently. And that's not because that error happens very frequently during sequencing. That is because of this process of RNA editing going on in cells. So the presence of A to G changes in RNA-seq data is evidence of editing at those positions. And so that allowed us to computationally analyze our RNA-seq data and to see how editing might be changed in our ADAR mutant mice. And so what we found is when we looked at the number of edited sites that with our depths of sequencing that we were able to detect, then actually, if anything, that was slightly increased, although that was not significant in the mutant mice. So it didn't seem that there was sort of a global defect in that regard. And we also looked at the percentage of editing at each site. And so these are plots where the black line here shows the median, and then you've got the spread of the data. So you can see that some sites are really highly edited and other sites are not very highly edited. And the average is about 10%, and that was exactly the same in our mutant animals. So it didn't seem that there was a global defect in RNA editing. However, what we were able to find is a smaller subset of sites, and that was around 8% of all of the sites that we detected, around 3,000 sites, where editing was largely normal in our wild-type mice, but was very profoundly reduced in the mutant mice. So what we believe is that there is a subset of about 8 or 10% of RNAs that ADAR1 edits, where the Z alpha domain is really important for recruiting and recognizing this RNA for editing by the deaminase domain. And so that then led us to conclude in the paper that we published last year that recognition of zRNA by ADAR1 limits interferon responses, as shown in this model here, where we've got ADAR1 P150 binding to a bit of zRNA through its Z alpha domain, and then the deaminase domain can change A to I, and that then prevents recognition by MDA5 and signaling through MAVs to induce interferon responses. And as I've explained, this conclusion is based on these three sort of principal observations. Those mice where the Z alpha domain is mutated such that it doesn't bind to zRNA spontaneously induce type I interferons and interferon stimulated genes in multiple organs. This interferon response required MAVs and protected against flu. And then finally, we found that about 8% of RNA editing sites that are usually recognized by ADAR1 require the Z alpha domain. So coming back to the question that I posed in the introduction about what the biological function of zRNA might be, we proposed that, that one of the functions is type I interferon induction via MAFs, and that activity is under normal circumstances limited by editing through ADAR1. Now, similar results were published by several of my colleagues in the field, and this includes these different papers down here, which all came out back to back at the same time. And going forwards, there are really a number of interesting questions that we like to address in the future. One thing that we don't understand, unfortunately, at the moment is what's unique about these 8% of editing sites that are recognized by ADAR1. We've not been able to detect specific sequence motifs or locations in the genomes or other things. And we really hope that through some more work, we'd be able to find out why these sites are recognized by ADAR1 through the Z-alpha domain and what's special about them. So that's something we don't know at the moment. And the way we think we might be able to answer that is to first understand the answer to the second question. And the second question is about the cell types in which those zRNAs are generated. So most of our work that I've presented was done in using RNA extract from lung. And obviously the lung is a complex organ with many different cell types, immune and non-immune cells. And we really like to dissect in which cell type ADAR1 is important to prevent an interferon response. I think once we've got that cell type, then we can do RNA-seq and editing analysis on that cell type, and hopefully that might guide us to understand better what those sites are and what their properties are that require the Z-alpha domain for editing. And then finally, I think there's some really interesting implications from a more translational perspective, in particular, about inducing interferon responses in cancers, which might synergize with checkpoint blockade. And so we are sort of exploring the idea that inhibitors to the Z-alpha domain might be used to induce an interferon response in cancers. And just to wrap up, I wanted to sort of show this cartoon as a summary of what I've shown you of zRNA engaging both ZBP1 to induce programmed cell death as well as ADAR1, and that prevents activation of an interferon response through MDA5. And there are really some interesting links to disease, and I think really in the context of cancer that might be most attractive and most important. And so cell death and interferon inductions are things that might be good to induce in tumors in terms of killing the tumor and in terms of activating an innate immune response that might synergize with adaptive responses, which could be unleashed with checkpoint blockers. And indeed, there's already some literature on ADAR1 showing that ADAR1 confers resistance to immune checkpoint blockade. So if you take ADAR1 out, if you knock it out from tumor cell lines in mouse studies, for example, then those tumors can be targeted much better by checkpoint blockers if they lack ADAR1, because they've got a bit of an interferon response going on, and that activates innate immune responses, which then will facilitate adaptive immune responses. And so as I've mentioned in the last slide, one thing that I'm quite interested in exploring is whether inhibition of the ADAR1 Z-alpha domain could induce moderate interferon responses in tumors for the benefit of tumor immune responses, and vice versa. There's some really interesting work that came out very recently from Alan Herbert's lab and Sid Balachandran's lab in this study here, where they've discovered a chemical compound that stabilizes zRNA, and they use that to induce ZBP1 dependent cell death in tumors to kill tumor cells. So if anyone's interested, I really recommend looking at that. And with that, I'd like to finish up. This is my group at the moment, and I'd like to particularly acknowledge my funders. So that's the Medical Research Council and the Wellcome Trust, primarily in the UK, as well as the Lister Institute, as well as people who've been in the lab in the past, and particularly Jonathan Mulfate, who's done the work on ZBP1 that I briefly introduced, and Kalika Tang, who has done the work on ADAR one, which I've presented. And I'd also like to thank our collaborators, in particular George Cassiotis and George Young at the Crick Institute in London, who've really helped us very much with the computational analysis of editing. And I'll stop here and I'll leave this on, which is the graphical abstract from our paper, which hopefully summarizes what I've told. And I'll leave this on whilst I'll take any questions that you might have. So thanks again for having me, and I'm very happy to discuss.
Thanks a lot, Jan. That was a wonderful paper. As I said before, I never thought I would ever have to read about zDNA until today. So it's really cool to just make sense of the information. And just a comment. This is a very nicely drawn graphical abstract. Who drew it? Was it the first author?
And that's myself. So I used Adobe Illustrator to draw it.
Oh, nice. This is like this. The color scheme and everything looks pretty cool in this one.
Yeah, lots of people use BioRender these days, but I think I've got a license for Illustrator, and that works quite well too.
Oh yeah, likewise. I love Illustrator. There's something about matter of pride whenever I try to move to BioRender. I think there is no way I can use that. I want to build all my graphical abstracts in Illustrator. I didn't learn this software for nothing. Okay, I had a minor question. So
sure.
There's another deaminase that I was aware of before this, is the AID that B cells use to hypermutate, right, for the DNA and make variations. Is there any? Do you think is this enzyme capable of making mutants of RNA? Adding those substitutions.
So I'm not an expert in AID and somatic hypermutation, but my understanding is that AID would not change RNA.
Right. It does. Yeah, that's the DNA thing.
ADAR would not act on DNA. So I, to the best of my knowledge, AID I think it says specificity for DNA.
Okay. Okay.
I mean, it's an interesting point whether that's got any, whether that might have any impact on recognition of DNA by CGAS.
Okay. Okay. All right. Anybody else got any questions?
So maybe one question about this elevated type I interferon response. So can it have any impact like on the autoimmunity?
Yeah, so in our mice, they don't develop autoimmunity, so they're viable, fertile, and my colleague Jonathan Mulfate, who's also studied these animals, has aged a cohort, and that's in his paper. And other than some minor differences in weight, there's really no signs of sort of a full-blown autoimmune response. Now, interestingly, the same has happened with other knockout mice that are meant to mimic these types of interferon-driven disease in human. It's similar, for example, with the SAMHG1 knockout mice, SAMHG1 being another gene that, when mutated in human, causes interferon-driven autoinflammation. If that's knocked out in the mouse, these mice again are fertile and viable. They've got an ISG response, such as these ADAR mutant mice. And on a speculative note, what we think might be going on is that these animals live in a very clean animal facility in a specific pathogen-free cage, and that in human, the mutation goes along with some environmental insults, such as infections that people might have, and that then there's a more profound response, and then the autoimmune disease eventually precipitates. So that's one potential explanation, but of course there might be mouse-human differences as well, that there might be different sets of interferon-stimulated genes in mice and humans, and that might explain why similar mutations or loss of functions cause a disease in human, but they don't cause in mice.
Okay.
Thank you, and thank you for a great presentation.
Thank you.
This is the last one for myself. what's the next topic you guys are working on right now?
Yeah, so what we're really quite interested in is whether there's a specific cell type in which this is going on.
Okay.
And what we've got in the paper, it's sort of in the middle of the paper, and one of the figures in the middle is some sort of circumstantial evidence that neutrophils in the lung might be important. And the reason for that is that if we look at interferon stimulated genes, then these go up in lots of different cell types if we sort them from the lung. But there are certain interferon stimulated genes that go up in neutrophils, but that don't go up in other cells like B cells or T cells. And interestingly, these interferon stimulated genes that go up in neutrophils, but not in other cell types, they can be induced not only through signaling through the interferon receptor, but they can be directly activated through IRFs. And so the IRFs are the transcription factors downstream of MDA5 MAPs and other nucleic acid sensors that induce type 1 interferons, but they also induce a subset of interferon stimulated genes. And so we think that maybe that information is evidence that neutrophils might be an important cell type in which this response is initiated. And so what we're doing at the moment is we're trying to find evidence whether that's true, or whether another cell type might be the cell type in which this is happening. But the idea is that this is happening in some cells, and then they make interferon that acts in a paracrine way on other cells. And so we're trying to first define sort of a gene set that would signify activation of MDA5 MAPs. And then with that, we're trying to find the cell type and then do some editing analysis in that cell type. Now that all sounds a bit complicated, because you probably think, oh, why don't you just measure interferon in the different cell types? And the reason for that is that interferon, type 1 interferon, is very, very hard to measure, both at mRNA and particularly at protein level, because both mRNA and protein have a very, very short half-life. And it's very, very hard to pick it up by RT-qPCR, and it's even harder by ELISA. And you'll only pick it up if there's really a lot of it being made. And in this setting, with the mice being healthy, they make some interferon, but not tons like you'd have in an acute virus infection. And so therefore, we need to have some sort of surrogate readout other than just looking at interferon, because it's just below the level of detection.
Yeah, that sounds like a good plan for the next few years. And also, I like what you just what you mentioned about short life of interferons and mRNA. I just realized maybe that's a feature, right? That you don't want to have a prolonged response. The response stops as soon as the trigger stops.
Yeah, so you've got, I mean, there's lots of work on this regulation of mRNA half-life, cytokine mRNAs, and some of them, including interferons, have in the 3' untranslated region, so that's a bit on the mRNA behind the stop codon. They've got so-called AU-rich elements, which are basically stretches of sequence that are rich in A and U's. And these in turn are recognized by RNA binding proteins that then target the RNA for degradation. And so many cytokine mRNAs have these are related RNA elements that make sure the RNA is really short-lived, such that you get a very short burst of the cytokine to shut it down afterwards. And that's probably a mechanism to prevent prolonged inflammation.
Yeah. Oh, yeah. That makes a lot of sense. Okay, Jan. Thanks a lot for joining us here today and talking to us about this wonderful work. I think I'll not forget several of the points that I learned in today's presentation. And every time my family has a dinner party conversation about zRNA, I have something to talk about now specifically. I
Great.
never
Thanks
knew
for having
anything
me.
about zRNA. But yeah, Jan, thanks a lot for having us. And for our audience, thanks a lot for joining us for the Journal Club. We'll be back soon. And until then, we'll see you later. Bye bye.